Hi everyone, it's Gigi, and in this video I'm going to be discussing how DNA acts as our genetic material within living things. But before we knew that DNA made up our genetic material, there was a controversy in the 1900s where scientists debated on whether or not our genetic material was made up of DNA or protein. And so this big question in the 1900s is what sparked the idea for the following experiments. But more specifically, I'm going to be focusing on Hershey and Chase's experiment. And right here is Martha Chase, and here we have Alfred Hershey. And so in the upcoming frames here, I'm going to give you guys some background info on Hershey and Chase, along with a brief explanation of some other experiments performed by scientists that were all looking to answer the big question of the 1900s, which actually helped Hershey and Chase in the long run to orchestrate their own experiment. So the background information that I'm going to share with you guys is just going to help you later on in this video when I'm explaining a bunch of the experiments. And so Hershey and Chase studied bacteriophages, which are viruses that kill bacteria. So since Hershey and Chase were so familiar with bacteriophages, they ended up using them in their experiment. And so Hershey and Chase were actually the last group of scientists that experimented, and they were just looking for the genetic material. I'm not going to write it all out, but you guys understand that we're talking about the genetic material. And so one of the first scientists that attempted in getting this answer to the big question in the 1900s was Frederick Griffith. And in 1928, he was working on bacteria called Streptococcus pneumonia. And so this Streptococcus pneumonia had two strains. One was smooth and the other was rough. And so the circle around the S here is symbolizing the protective capsule. And this protective capsule protects the cells from the immune system, which makes this a pathogenic cell, while the rough does not have the capsule layer for protection. It makes it non-pathogenic. And so then Griffith decides to inject the mice with each strain. So one mouse had been injected with a smooth strain and the other was injected with a rough strain because he wanted to see the effects of the different strains of Streptococcus on the same type of model, so the model being the mouse. And so with this, the mouse that was injected with the S strain died. And so with that, Griffith then performed a second experiment. And in this experiment, Griffith took heat-killed cells from S strain, so it's going to look like this. And then he added it with a rough strain. So now he's putting both strains into the same mouse. And the result was only S colonies and no transformation principle. So the transformation principle would have occurred right here. And the transformation principle is what would make the R into an S so that the total production would come out to be S colonies. So with Griffith not being able to find the transformation principle, then came Avery, McCarty, and MacLeod, which worked together in 1944, and they were still looking for the transformation principle that Griffith could not find. And so I'm going to show that experiment in the next frame here. So Avery and his colleagues, they ran a set of experiments in which they took cell lysate of heat-killed S strain and treated it with different enzymes, like you can see all under this column right here. And here is our heat-killed S strain with our lysate. And so it's going into each enzyme here. So after the treatment with the enzyme has been properly carried out, it's then mixed with a rough strain, which is going to go with every enzyme. And then it gets properly incubated, and that's when we see our results. So here are the results. As you can see here, most of them are S colonies, only because the rough strain somehow was converted into the R, which is kind of what we saw in Griffith's experiment. But right here, with the DNase enzyme, you can see how it's an R strand. And so that is our result that we were looking for. So we've noticed that DNase was the only treatment that failed to convert the R strain into an S strain, which meant DNA was the transformation principle. But Avery then realized that he performed this experiment all outside of a living system, so it wasn't really what they were looking for because the question referred to within living systems. So they're still looking for an answer. So comes along Hershey and Chase in 1952. So Hershey and Chase began their experiment by labeling their bacteriophages by letting them grow in a radioactive molecule which gave them a phage coat. And so this first experiment phage here was labeled S35, and it was a protein coat. And so that protein coat is going to be shown here. This is your signal. It's like almost like an indicator. And it's going to be green in this case. And so after the phage is labeled and the coat is on, it's going to then infect the bacteria. And so here are the phages right here, S35 phages, infecting our bacteria with our coat on. And so after that, it's then going to be put into a blender. And so it's going to have everything in there. So it has our coat, our phage, and the bacteria. And so the blender is going to separate the phage coat from the bacteria. And after that, it's going to be centrifuged in order to give us this pellet-like structure here. And that's really what we're analyzing, well, what Chase and Hershey analyzed in order to indicate what the genetic material was. And so you could see the green protein coat all along right here, and that is called the supernate. And that's where the virus is. So the supernate contains the virus, and it's containing our protein coat right now. And the dense part right here at the bottom, circle it, is this yellow, and that is our bacteria. And it kind of just sits at the bottom underneath the supernate. And with our green coat being in the supernate, that gave us our result that protein cannot be the genetic material since it can't get into the cell when there is an infection. So with the first experiment showing that protein can't be the genetic material, then came the second experiment in which they labeled DNA. And so here's our DNA coat on our phage. It's going to be red this time instead of the green. And again, this is the signal we're looking for, and we want it to be in that bacterial cell. So we want it to be somewhere down here at the end. So they let the phage grow in the radioactive model, and they label it as P32. And so then it goes, infects the bacteria with the coat on. Draw that quick. And so as the DNA is infecting the bacteria, it's seeping into it. So this is it really infecting it. Then all the remains go towards the blender. We have our bacteria, our phage, and our phage coat, which is red because it's P32. It's our labeled DNA. With that, we put it into the centrifuge to get it to look like a structure much like this pellet. And the results here, I didn't want to show them before, so the results looked like this. So you could see here that our supernate is technically just our bacteria. And right here, the bacterial pellet is our DNA. So that's going to give us our conclusion, which is that DNA is in the bacterial cell, which indicates that this is the transformation principle because DNA can now go into the cell and create new viruses, and that will change it forever. So now that we've gone through Hershey and Chase's experiment, both of them, and we've gotten our answer, we know what our genetic material is, and we've seen how they've proven it, we can now talk about the importance of all this. So our first question is asking why scientists were even researching this. And so they were obviously researching it because they knew genetic material is what is passed from parent to offspring. So much like that. And it also was further researched because of the big question in the 1900s when people questioned what our genetic material was. And so for our second question, what are the functions of genetic material, which we now know is DNA? So the first function, like I mentioned before, is how traits are inherited. And so inside an organism's DNA is your genotypes, which are clusters of genes within the body. So you can't physically see a genotype trait, but you can see the opposition of that, which would be a genotype. So down here I have brown hair phenotype. And so those are going to be located on the chromosome, which is inside the body, so you obviously can't see that. And so that would transfer over into the physical symbolization of it, which would be your genotype. And so that's where your red hair comes in, along with the brown hair over here. So they're physical traits that you can see. And so the other function that DNA does is it basically runs protein synthesis because it creates an mRNA so that the mRNA can then take coding from the DNA to create protein, which is what protein synthesis is. It's the production of proteins. So DNA is extremely essential to the body. So our last question is more bias, and it just asks why I chose this for my bio final project. And I chose this because I simply enjoy learning about genetics. But more specifically, I chose to do a video format and this topic because I knew I could then translate each experiment in a way that I can illustrate it and understand it better myself. And I understand it so much better that I even noticed that scientists then started bouncing ideas off each other, much like how Avery and his colleagues in 1944 tried to find that missing transformation principle that Griffith couldn't. And so I hope this video really helped to break down the experiments for you guys and learn how we came to know that DNA was our genetic material.